Hi guys, Miss Francis here. Last video we discussed the sodium-potassium pump, which is a form of active transport. Now I'd like to explore ion pumps. Ion pumps generate voltage across membranes. All cells need to maintain a voltage difference, also known as a charge difference. So this is what I mean. A voltage difference can also be called a membrane potential, and it's that sodium-potassium pump that we last spoke of that helps maintain that membrane potential, otherwise known as the voltage difference or the charge difference, where that membrane potential acts like a battery. It favors the passive transport of cations, positive ions, into the cell, and anions, negative ions, out of the cell, where two combined forces, collectively called the electrochemical gradient, drive the diffusion of ions across the membrane. So here's what we can write: that cations are positive ions that move into the cell, or are favored to move into the cell. Anions are negatively charged ions, and movement of anions is favored out of the cell, where the electrochemical gradient is due to two forces: the concentration gradient, in other words, the chemical force, whether I'm going from high to low or low to high, and the electrical gradient, also known as the electric force, which is based on that membrane potential, based on that charge difference. Now, the sodium-potassium pump is crucial to the functioning of neurons in the nervous system along the axon, because what that sodium-potassium pump does is it restores that membrane potential. This is what we're going to be talking about in our next video. Proton pumps help to create electrochemical gradients. In plants, bacteria, and fungi, it's this proton pump that's the major electrogenic pump, actively transporting hydrogen out of the cell. Now, these proton pumps are going to be vitally important in respiration and in photosynthesis. So in the mitochondria, when they're used for photosynthesis, for respiration, those proton pumps are going to be found in the cristae, and as far as chloroplasts go, those proton pumps are going to be found in the thylakoids. In both the mitochondria and the chloroplasts, the proton pumps are going to store energy that can be used for cellular work. Proton pumps can also be found in the human stomach, and it helps create the highly acidic environment, because the more hydrogen ions that I have on one side, the more acidic the environment becomes. Co-transport also involves an energy or ATP powered pump, but the way co-transport works is slightly different from our other pumps. So let's take a look at it. Basically, in co-transport, it's going to occur in two steps. So the first step is that ATP is going to be used to transport the solute. So let's look here at the green. The first step is that ATP is going to be used to actively pump hydrogen, in this example, through a proton pump out of my cell. In the second step, that hydrogen is going to diffuse back down its concentration gradient, because inside the cytoplasm I have a low concentration of hydrogen. Outside of the cytoplasm I have a high concentration of hydrogen. So that hydrogen naturally wants to go back into the cytoplasm, but it requires the help of a membrane protein, so it's going to diffuse back via facilitated diffusion. Now, as that hydrogen diffuses back through, it's going to carry with it sucrose. However, the sucrose is going to be actively transported through this co-transporter. Now, I don't see any ATP over here by the sucrose. So where does the energy come from that moves that sucrose against its concentration gradient? Well, the movement of ions are providing the energy. So in this example, it's the movement of the hydrogen ions that are going to drive the sucrose across the cell membrane. Here's an example of how this would apply to us, to humans in real life. Let's say I eat a bagel and I break down the bagel into glucose. But now I want to get that glucose through my small intestine, which absorbs nutrients such as glucose, and then into my bloodstream so that it can be transported throughout my body and go where it needs to go. So here I've got the microvilli, which are the finger-like projections of the small intestine, which help increase surface area, enabling more glucose to pass through these epithelial cells, which are the cells that are in the lining of the small intestine, in order to get through to the bloodstream. So let's see how that might happen. Well, the glucose is going to get moved into this epithelial cell by co-transport. And since we're talking about glucose, its co-transporter is sodium. Sucrose goes with hydrogen, glucose goes with sodium. So how are we going to get the glucose from that small intestine into the cytoplasm of the epithelial cell? How is it going to do that? Co-transport with the sodium. And where does that energy come from? There's no ATP. The energy comes from the movement of those sodium ions. Now, how is this concentration gradient maintained? It's that sodium-potassium pump. Once again, that plays a vital role in maintaining this concentration gradient. The last type of transport that I want to talk to you about today is called bulk transport, and bulk transport involves exocytosis and endocytosis. So what are exocytosis and endocytosis? Well, exocytosis enables large molecules such as polysaccharides and proteins to cross the membrane via vesicles. These transport vesicles are going to move to the plasma membrane, fuse to the plasma membrane, and then spill the contents to the outside of the cell. So now I've had my polysaccharide or my protein exit the cell. On the other hand, we've got endocytosis, which brings in large molecules such as polysaccharides and proteins. So endocytosis is a reversal of exocytosis. So how does this happen? Small area of the plasma membrane sinks inward to form a pocket. Then that pocket pinches in, forming a vesicle containing the material that was once outside of the cell. It's also known as cellular eating, and the contents of the vacuole are then going to be digested when the vacuole fuses with a lysosome. Pinocytosis is also known as cellular drinking, where a cell creates a vesicle, but this time it's around something such as a drop of extracellular fluid. So let's compare and contrast these three types of bulk transport. Endocytosis includes phagocytosis, pinocytosis, and we haven't gotten to it yet, but includes receptor-mediated endocytosis. So here's phagocytosis. I am eating by taking in a solid particle. Pinocytosis, I'm drinking by taking in some sort of fluid. Now let's talk about receptor-mediated endocytosis. So this process is triggered when an extracellular substance binds to what's called a ligand. This ligand is a receptor. It's going to receive that substance. And the ligand itself, the receptor, is located on the membrane surface. And what that does is triggers the formation of a vesicle. An example of receptor-mediated endocytosis is how human cells use this process to absorb cholesterol. So this ends our discussion on the cell membrane and cell transport. Our next unit is going to look at the neurons and our kidney.